I'll invite our next speaker up to the podium, Dr. Ben Blencowe, one of our stars. Many folks internationally are aware of his seminal work in defining the complexities and the nuances of the splicing mechanisms discovered first by Dr. Sharp, as you all are aware. I have a small but warm story. Not only is he a brilliant scientist, but he's a wonderful fellow. I remember 10 years ago he helped with one of the graduate students studying the splicing of dicer RNA. And I remember this was a feisty, bright student, and in his defense, he decided to take on Dr. Blencowe a little bit on the splicing machinery, which I thought was a little bit like someone taking on Moses with the 10 Commandments. So let's invite Dr. Blencowe, up to talk today.
Thank you for that very generous introduction. Phil, it's really a great honor and pleasure to be invited to this symposium. I'd like to thank John Dirks and his colleagues at the Gairdner Foundation for the invitation. And what I want to do today is to tell you about work we've been doing directed at the global scale discovery and characterization of alternative splicing regulatory networks. I'm actually going to start by giving a sort of overview of some key questions in the field, and then I'll transition to how some recent work has led to the illumination of a landscape of microexon regulation in the mammalian nervous system, and how this work has provided some interesting new insights into the mechanistic basis of neurological disorders, in particular autism spectrum disorder. So Lynn has already introduced this yesterday, but just to remind you that one of the major surprises to emerge from different genome sequencing projects is that we have a comparable number of protein coding genes as far less complex species. So C. elegans, shown here, is an organism that comprises approximately 1,000 cells, and it has a similar number of protein coding genes as humans, and we have hundreds of trillions of cells in our nervous system alone. So this begs the question of what drives organism complexity and species complexity, and you've already heard today that there's an expansion of non-coding RNA regulation during evolution, and this is one of the answers. But alternative splicing plays a very important role in this process. And this slide just summarizes some of the different types of alternative splicing events that we're familiar with. So cassette-type alternative exons, which are spliced in and out of messages, is the most frequent class found in mammalian cells and tissues. There's alternative 5' and 3' splice site selection events, more complex events, for example, involving mutually exclusive alternative exons, and then intron retention, as it turns out, is also a frequent process of alternative splicing in mammalian cells and tissues. Is also very frequent in plants. And as everybody here, I'm sure, is already aware of, these different classes of alternative splicing can produce distinct mRNA and protein variants. But also, as Lynn mentioned yesterday, and thanks to Lynn's seminal work on NMD, we understand that alternative splicing can also introduce premature termination codons, either when exons are included or in some cases when they're skipped, to trigger the process of nonsense-mediated mRNA decay, and this plays an important role in controlling gene expression. And we think it's important for approximately 1/3 or more of the regulation of 1/3 or more of human genes, in particular through intron retention. So when we look at the proportion of genes that produce alternative splicing variants, this is just showing some very rough estimates from using high-throughput RNA sequencing. Sandy Pan and colleagues in my lab, together with Eric Wang and Chris Burge's lab several years ago, showed that over 90% of human protein coding genes generate alternatively spliced transcripts, and we're aware that in these other species the frequencies are less, there are fewer in number. But what's more informative is actually to look at related genes between different species. So if you just take two homologous genes and you sample the frequency of alternative splicing between these, again, we can use high-throughput RNA sequencing to quantify this, where we're mapping reads to the junctions formed by splicing or alternative splicing. And when we plot the frequency of exon skipping between different vertebrate species, in this case, we find that indeed there is a higher degree of alternative exon skipping in humans and non-human primate species compared to these other vertebrate species, and there's this gradual decline in the frequency of alternative splicing. So we think it plays a very important role in driving organism complexity, and if we look at the literature is full of examples of really important alternative splicing events. And just to name a few here, the first example of this, which came 3 years after Phil Sharp and Richard Roberts' papers showing the discovery of splicing, the first example from the Baltimore and Hood groups show that alternative splicing can control the switching between membrane-bound and soluble forms of IgM. And then a few years later, Bruce Baker and colleagues and others show that there's a cascade of alternative splicing events that control sex determination in Drosophila. Work from Zipursky, Brent Gravely, and others has shown that the DESCAM gene in Drosophila has the capacity to generate 38,000 different mRNA isoforms. And these isoforms, which come from the DESCAM gene, play a critical role for wiring of the Drosophila nervous system. And then work from my own group, spearheaded by Mathieu Gabou and Hong Han, has shown that a single alternative splicing switch in a transcription factor known as FOXP1 can control the transition between embryonic stem cell pluripotency and cell differentiation. It does this by changing the specificity of this transcription factor. And this is actually occurs in other transcription factors as well. So splicing can control entire transcriptional programs during development. Despite these examples, we really know nothing about the function of the vast majority of alternative splicing events associated with normal and disease physiology. And this gap in knowledge is especially relevant to the nervous system, since it possesses among the most complex repertoires of splice variants. So to address these questions, we've been, over the years, developing and applying different high-throughput approaches and trying to improve on computational pipelines for detecting and quantifying splicing. And the work I'm going to describe to you is largely spearheaded by Manuel Remia, a former postdoctoral fellow in the lab who's now a group leader at the EMBL CRG Institute in Barcelona. And a lot of the work was also done by Ulrich Braunschweig, as well as other people in the lab, which I'll come back to at the end during the acknowledgements. So to develop this pipeline, what Manuel did is to assemble a comprehensive repertoire of splice junction sequences mined from RNA-seq data, and his pipeline can detect these different classes of splicing events, which I already summarized to you at the beginning. But a more unusual feature of this pipeline is that it is also set up to search for the usage of previously unannotated exons that lie within annotated introns. And I'll come back to how this has allowed us to discover numerous new very short exons, which we call microexons. This just shows a heat map where we're looking at the profiles of alternative splicing mined using the pipeline I introduced on the previous slide. And the scale here indicates a metric known as percent spliced in. This is the percent of transcripts that have an exon spliced in, and blue indicates where there's more exon or intron skipping, yellow, more inclusion of a sequence. And you can see that there's these strikingly different patterns across 50 different human cell and tissue types. And then nervous system tissues cluster very cleanly at the left hand of this heat map. And this, again, represents all classes of alternative splicing I showed you on the previous slide. Now, when we look at the genes that concentrate these alternative splicing events, there's about 2,500 of them. We find that they're highly enriched in functions that are important for nervous system development and biology. So there's many genes involved in neurogenesis, axonogenesis, synapse functions, vesicle transport, signaling processes in the nervous system, ion channels, and so on and so forth. One of the, I think, most striking findings from this mining exercise was the discovery of this set of alternative microexons. And just to show you in more detail how Manuel discovered these, again, what he did was to simulate all possible joinings of 5' and 3' splice site sequences within annotated introns, separated by as short as 3 nucleotides and going up to 27 nucleotides in length. And then he asked which reads from the different cell and tissue types that I showed you in that heat map align to the sequences that are formed by the inclusion of these very short simulated exons. So we're looking for reads that support the existence of these exons. And after various filtering steps, what he found is that there are about 250 alternative microexons detected in human and mouse. He surveyed a similar number of human and mouse cell and tissue types to look for conservation. And a striking observation is that approximately 65% of these alternative microexons are conserved between human and mouse. And this contrasts with approximately 29% of longer alternative exons. And moreover, among the conserved alternative exons, 75% display very striking neural specific inclusion, both in human and mouse, and this contrasts with about 27% of longer alternative exons that show neural inclusion. This conservation at the level of regulation is reflected by sequence conservation. If we compare sequences across mammalian genomes, we find that the sequences both within and surrounding the exons in the intronic sequence are more highly conserved for the microexons, including microexons as short as 3 to 15 nucleotides, relative to longer alternative exons and constitutive exons. What's more is when we look at the frequency at which exons of different length are found, we find that the microexons show very strong selection pressure for preservation of reading frame. Most of these exons are located in encoding sequence, and you can see here if we just count the number of alternative exons of different length, you see this very strong bias for having multiples of 3, and this relationship breaks down as you sample longer alternative exons. And again, most of the exons in the short range here show neural specific inclusion. So this just shows 4 representative examples of these exons when we analyze RNA-seq data across a time series of neuronal differentiation from ES cells. You can see here at a glance that these exons show this striking increase in inclusion, and this actually applies to most of these exons. So 75% of the neural-regulated microexons have a percent spliced-in switch of at least 70% during neuronal differentiation. So what are the functions of microexons? This is work that was largely spearheaded by Robert Weather, a postdoctoral fellow in the lab, and what he showed is that the amino acid residues encoded by these microexons are predicted to be largely surface accessible. They're enriched in charged amino acids, and we also find that they're enriched in protein interaction domains, so sequences encoding protein interaction domains. And this contrasts with what you find for longer alternative exons, which tend to be excluded from folded protein domains. And when we look at tertiary structures, where we can actually map the residues into existing structures or model the location of these exons on existing structures, we find that in every case, and this just shows 4 examples, but we've seen this for dozens of others, these residues are almost invariably on the surfaces of proteins, or sometimes they're located on the loops of loops that project out from these protein structures. And here's an example of a microexon that's located at the interface between 2 interacting proteins, CDC42 and DOC9. So these properties suggest an important role in mediating or controlling protein-protein interactions, and to test this, we focused on a few examples, and this is work from Jonathan Ellis and Dave O'Hanlon in the lab in collaboration with Mariana Babor and Fritz Roth. We modeled the function of this 2 amino acid microexon in a protein called amyloid precursor binding protein 1. The 2 residues were predicted to interact with a binding partner Cat5, and when Jonathan and Dave deleted this exon using a luminescent protein-protein interaction assay, we found that there's about a 2-fold drop in the interaction with Cat5 when this particular microexon is deleted. We can confirm that by co-IP Western blotting experiments, and we see a significant drop in the interaction with this particular binding partner. So this is a relatively subtle change in interaction. Other examples that we've looked at, including a microexon in the adapter protein 1 subunit S2. When this microexon is deleted, we see an almost complete loss in the interaction with another subunit of the AP1 complex, known as AP1B1. So if you're wondering what AP1 is, it's an intracellular transporter, and interestingly, mutations in this protein have been linked to autism spectrum disorder as well as X-linked mental retardation. And this was a sort of recurring theme in the set of genes that contain these microexons, since we found many other examples where microexon-containing genes that should display these properties have genetic links to autism. I'll come back to this in a minute. So these questions or observations raise this question here, which is, are microexons misregulated in autism spectrum disorders? Just as a few words of introduction to this disorder, it's characterized by cognitive impairment, impaired social interaction and relationships, repetitive and stereotypical behaviors, impaired language and communication skills. It's prevalent in the population. It's found in at least 1 in 88 individuals, and it's more common in boys compared to girls. And a challenge in this area, this field, is the heterogeneity of this disorder. So there's been at least 1,000 genomic mutations and other variations associated with ASD, but few are common among even small subsets of affected individuals. So the question is, to what extent do ASD-associated genetic alterations lead to convergent effects on specific pathways? And this is a question that we became interested in several years ago through a collaboration with Dan Geschwin's group at UCLA. Dan's group had access to brain samples from deceased autism individuals, and he used microarray profiling to characterize the transcriptomes of these samples together with a similar number of control samples. And what he found is that there is an altered gene expression signature in the brains of at least 1/3 of analyzed individuals with ASD. And we performed RNA sequencing on some of these samples, and we found that longer cassette alternative exons display altered regulation. We could tie this to the misregulation of a splicing factor called RBFOX1. So we decided to reanalyze these samples in addition to RNA samples from a larger cohort. And the striking observation here is that at least 30% of the detected microexons in these samples show misregulation that belong to this short length class here, so the 3 to 15 nucleotide class, show a misregulation of at least 10% PSI between cases and controls. We also see frequent misregulation of the longer microexons, but then only about 5% of exons beyond 27 nucleotides show misregulation across these samples. And as I already alluded to, when we look at the genes that display misregulated microexons, they're highly enriched in neuronal development and synaptic genes that are genetically linked to autism. So in red are some well-established genetic links, including genes such as SHANK, ANK2, ROBO, and in the shaded oval here are genes that have been genetically linked that are less well established. So given these observations, we wanted to next ask, what is the mechanism of neuronal microexon regulation and its misregulation in autism? So part of the answer to this is this factor here, which we call NSR100. This is the neuronal specific CRH-related protein of 100 kilodaltons. It's a protein that was discovered in my lab by John Colarico, former graduate student who's now a fellow at the Bauer Institute at Harvard University, and Bushra Raj, a former graduate student who's now a postdoctoral fellow at Harvard, characterized the mechanism of this factor. And what she showed is that through collaborations with Eduardo Iras, Sancloa Gingra, and Gairdner Ulu, is that this factor can bind very specifically to these UGC motifs located immediately upstream of 3' splice sites. And these sequences act as enhancers of splicing, so when this protein's bound in neuronal cells, it stimulates the assembly of early splicing complexes, leading to the excision of this upstream intron, and this allows inclusion of these exons. So given the insight into the mechanism involving NSR100, we went back to the autism patient samples to ask the question, is there a correlation between NSR100 expression and changes in microexon inclusion across these samples? So this is representing microexon inclusion as a z-score across the different ASD samples and control samples. And in the blue line here, you see the level of NSR100, which we're tracking in the same plot. And we find that there's indeed a significant correlation between the mRNA expression of NSR100 and the inclusion of these microexons across the cases shown in here in red and the control samples. But this is really just a correlation. So we then wanted to address, does NSR100 deficiency play a causative role in autism? We don't have the full answer to this, but we, through a very productive collaboration with Sabine Cordes' group in developing a mouse model, we're beginning to obtain some very interesting insights into this question. And this is work that was spearheaded by Mathieu Quesnel-Valliere, a graduate student in the lab who's co-supervised by Sabine. I'm not going to go into the details of the targeting construct that we use, but suffice it to say that Mathieu deleted exons 7 and 8 in the NSR100 gene. This leads to loss of full-length expression of NSR100, as well as loss of expression of the protein in the null mutants. The heterozygous mutants express about 50% normal levels of NSR100. So Mathieu did a lot of work to characterize the null mutants. Most of these mice die at birth. There's 85% perinatal mortality, and there are numerous neurodevelopmental defects in these mice, including misregulation of neurogenesis timing. This leads to cortical layer thickening, which by the way has been seen in post-mortem samples from autism individuals. There's impaired neurite growth in neurons that we cultured in vitro from these mice, and they also display midline crossing defects. When we look at microexon regulation, we see something similar to what we see in human subjects, in that the microexons that we've analyzed, I'm just showing 3 examples here, but we've performed RNA-seq analysis on these samples, and we see that there's widespread loss of microexon inclusion, with the hets displaying intermediate levels of inclusion between the wild type and the null mutants. So the hets initially didn't display any obvious phenotypes. And then Mathieu started running them through various tests, including locomotion, habituation, anxiety, short-term memory, olfaction, hearing, and vision, and they essentially look normal in these tests. So then we wanted to ask, how do they behave in social behavior and other social behavior tests, and what do they look like in terms of other features of autism seen in patients? So one of the tests he performed is assessing sociality with this reciprocal interaction test, and what you do here is to ask simply, do wild-type mice spend more time interacting with each other compared to the mutant mice? And you can see at a glance here that there's a significant difference in that the mutants, the het mutants, spend less time interacting with each other than the wild-type mice. But a more sophisticated test for this is this 3-chamber apparatus, where we can assess social choice as well as social novelty. And the way this works is that you place a mouse in this 3-chamber apparatus, and you ask how much time does it spend with a stranger mouse compared to an inanimate object in the first test. The second test is to ask how long does a test mouse spend together with a stranger mouse compared to a familiar mouse. And just to sort of show you what this looks like, click. Sorry about that. You can tell I'm not a PC person. Yeah, so here's a wild-type mouse moving around the cage, and wild-type mice are curious. They're social animals. It's spending time with a stranger. Pretty soon it's going to start exploring the cage. It's wandering off to check out the object. Pretty soon it gets bored of that. It's starting to gravitate back towards the stranger mouse. And this one is doing something similar. It's checked out the familiar mouse, and it will start moving back towards the stranger mouse. So wild-type mice will spend more time with the strangers compared to the object and the familiar mouse. But what happens with the het mutants? So this is a heat map that shows the amount of time that the het mutants spend in the 3-chamber apparatus compared to the wild-type mice. So red indicates more time with the hets, grayscale indicates more time with the wild-type mice, and you can see at a glance that the hets are spending more time close to the object, whereas the wild-type mice are spending more time, as expected, close to the stranger. So the hets prefer interacting with an inanimate object over a stranger mouse, and then in the social preference test, we find that the mutants prefer interacting with a familiar mouse over a stranger mouse, where they simply avoid social interactions. They're spending more time in the middle of the apparatus, and these differences are highly significant. So we've done a lot of additional work to look at properties of these mice, and so far they're recapitulating other hallmarks of ASD. One of these properties is we see increased density and altered morphology of dendritic spines. This is just comparing wild-type neurons in the heads. And there is also an imbalance in glutamatergic signaling that leads to a situation where we see spikes of similar amplitude as we see in the wild-type mice, but they're fewer in number, indicating that these are less mature synapses that are not firing in the correct way in these electrophysiology experiments. So with that, I'd like to summarize what I've shown you. So using a new RNA-seq pipeline, we've illuminated a landscape of conserved microexon regulation in the mammalian nervous system. I think an important observation here is that this misregulation is widespread. It seems to be affecting 1/3 of analyzed cases. These microexons are enriched in genes that display genetic links to autism. The level of inclusion of these microexons is significantly correlated with NSR100 expression, and then using a mutant mouse that is expressing approximately 50% normal levels of this protein, we find that it displays multiple properties of autism spectrum disorder, and we're excited by these findings since they suggest possible new routes to therapeutic strategy in the future. So I think I've gone through and mentioned most people have contributed to this work. So again, Manuel Arimia, Robert Weather, Jonathan Ellis, Dave O'Hanlon, and Thomas I didn't mention, but he's done a lot of work to validate the data that we've discovered using microarray profiling. Bashar Raj on the microexon project, Mathieu Canal Valliere, and Tim Sternweiler generated and analyzed the data looking at the behavior of the mutant mice. This work was performed in collaboration with Sabine Cordes, and we have enjoyed many fruitful collaborations with additional researchers here at the University of Toronto. With that, I'd like to thank you for your attention and welcome any questions.
Questions for Dr. Blencowe?
So there seems to be a link between ASD and long genes in neurons. I'm sure maybe you've thought about this before, but do you think that microexon splicing might be affected in a more severe manner in the long genes as opposed to ordinary genes?
It's a good question, and offhand, I don't think we've looked at that. It's an interesting question. So what I do know is that the introns that contain these microexons, as far as I recall from all the analysis that was done, don't display any differences, significant differences in length. If you plot the distribution of length of these introns harboring the microexons relative to other exons, there's no obvious difference in length, but it doesn't really answer your question, so that's something we could look at. Phil?
SR100, because of the SR, is probably controlled by a lot of phosphorylation and click and all the other things. Massive phosphorylation regulation.
Yes.
Do you see any genetic evidence that those kinases could be influencing this pathway? Are there mutations or variations in any of those kinases? Because
Not so
kinases can be targeted,
Yes.
obviously.
Yes. Not so far. It's a good question. With all the genomic sequencing data, that's something that should be looked for. I can say that with a collaboration involving Christian Schaff at Baylor, we've started to look at genotyping data, and as we discussed yesterday, there are mutations emerging in NSR100 as well as a paralog of NSR100 known as SRM3, as well as factors that we know are involved in the regulation of NSR100. So it's sort of emerging that this may be a point of not just regulation that we're seeing as I described it, but also a concentration of mutations in idiopathic cases of autism.